Hello, and today's lecture is on an introduction to energy, enzymes, and metabolism, chapter 6, reactions and energy. Going with chemical reactions, okay, the definition of a chemical reaction is the process in which one or more substances are changed into another substance. Okay, this focuses on metabolism, and metabolism is the sum of all chemical reactions that occur within an organism. Okay, this could be exergonic or endergonic, okay, as and chemical reactions. So exactly what is energy? Energy is the ability to promote change or do work. Okay, and in this unit, we're going to be discussing the two forms. Okay, the first one is kinetic energy, which is always associated with movement. And the next one is potential energy due to structure or location. And mostly we'll be focusing on chemical potential energy, which is the energy in molecular bonds as a form of potential energy, such as ATP. An example of kinetic energy by hitting the ball with the bat, and potential energy, which is on the arrow from the bow pulling backwards. This table that goes into different types of energy, such as light, heat, mechanical, chemical potential, and electrical ion gradient. Okay, as well as a description and example for each. The laws of thermodynamics, we'll be going over the first two. The first law of thermodynamics is the law of conversion of energy, which states energy cannot be created or destroyed, but can be transferred from one type to another. The second law of thermodynamics states transfer of energy from one form to another increases the entropy, which is the degree of disorder of a system. As entropy increases, less energy is available for organisms to use to promote change. And most of the times when we're talking about entropy, we're talking about heat being released. We also have to understand spontaneous reactions. Okay, the first one we have is exergonic, which is spontaneous, which means energy is released by the reaction. Okay, and it's important to note that this means negative free energy change. Okay, that's all the delta G stands for, is that free energy change. And then we have endergonic, which is not spontaneous. Okay, this is a positive free energy change, which requires addition of energy to drive the reaction. Quickly, we'll be going over this hydrolysis of ATP. Okay, what's important to remember is breaking this word apart. Okay, we have hydrolysis. If we break it apart, we have that hydro and lysis. Water, sorry, hydro meaning water, lysis meaning to break or to split. Okay, so when we're talking the definition of hydrolysis, this is the chemical breakdown of a compound due to reaction with water. Okay, this with is really important. It's not breaking water. It's breaking a chemical down with water. The energy liberated is used to drive a variety of cellular processes, which we'll be learning. See, when we add water, okay, to ATP, we have the hydrolysis here, which breaks that molecule apart. Okay, so instead of the three here that we have together bonded, we now have broken that apart with hydrolysis, and we have two on this molecule and one free phosphate. Move on to enzymes. Enzymes and Okay, an enzyme is a spontaneous reaction, is not necessarily a fast reaction. And so that's important to remember. We're used to the word spontaneous, meaning instantly, okay, but it doesn't necessarily mean a fast reaction. Okay, we have the definition of catalyst, which is an agent that speeds up the rate of a chemical reaction without being consumed during that reaction. So once it's used, it goes back and recycles itself. Enzymes are protein catalysts in living cells, as we've learned, and ribozymes are RNA molecules with these catalytic properties like enzymes. Okay, next, we're going to discuss activation energy. Okay, this is the initial input of energy to start a reaction. Okay, this allows molecules to get close enough to cause bond rearrangement. Okay, and it's common ways to overcome activation energies like large amounts of heat or using enzymes to lower the activation energy, which says positioning reactants together to facilitate the bonding. Okay, what this means is imagine yourself trying to get off the couch. Okay, that energy initially needed to push yourself off the couch would be activation energy. And here we just have a diagram that shows the activation energy needed to use a molecule or an enzyme. We're going to talk about lowering the activation energy. This is what enzymes do. Okay, so seen over here, we have the regular energy, and say that's the energy that you need to get off a couch. Then we have the energy with an enzyme. Say that's a friend helping and pulling you off the couch. Okay, you can do it and it requires less energy because that friend's pulling you off the couch. So that friend is acting like an enzyme. Okay, enzymes make biological processes happen faster than they would naturally. Okay, this will not work if pH or temperature changes. Okay, and that's important to remember. And substrates. An active site is the region on an enzyme where a substrate molecule binds and can undergo the chemical reaction. Okay, this is also the location where the reaction will take place. The substrate is the medium in which a chemical reaction takes place, the reactants that bind to the active site. And then you have the whole enzyme substrate complex, which is formed when an enzyme and a substrate bind together. Okay, the easiest way to imagine this, okay, is what we call a lock and key. Okay, so enzymes have a high specificity for their substrate. That means they have to have the exact key. Okay, so the lock and key metaphor for a substrate and enzyme binding, only the right key, which is the substrate, will fit in the right lock, which is the enzyme. This is called the induced fit phenomenon, which is the interaction also involves a conformational change. Okay, this conformational change means a changing in the physical property, but the easiest way to imagine this is a key fitting into a lock, much like a substrate into an enzyme. It fits perfectly and then can turn and change. Okay, so once again, that substrate is the key and the enzyme is the lock. We have an example. We see the substrate coming together on the active site. When that happens, we see that conformational change, okay, which is the color here, and it releases two different products. Now, what's also important to remember is this can also happen in reverse, okay? Same exact example, just with a more complicated enzyme molecule. Okay, and you could read yourself. Okay, the four different types of stages that happen in this complex.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
We're going over enzyme reactions. Okay, we have a term that's called saturation. Okay, saturation is the plateau where nearly all active sites are occupied by a substrate. Okay, we have two forms. We have competitive inhibition, where molecules bind to an active site. It inhibits the ability of the substrate to bind. Okay, so more substrate is needed. And we have non-competitive inhibition, where an inhibitor binds to an allosteric site not an active site. And I'll show a video here to you that'll explain this as well. For enzymes, we have these things called prosthetic groups, which are small molecules permanently attached to the enzyme. We have cofactors, which are usually inorganic ions that temporarily bind to an enzyme. And we have a coenzyme, which are organic molecules that participates in the reaction but is left unchanged afterwards. And here's an example of those.
This animation demonstrates how B vitamin coenzymes must be present in order to enable some enzymes to carry out their functions. Many of the B vitamins assist in the complex biochemical processes that produce energy from food. The B vitamins are essential parts of coenzymes. Coenzymes are part of the machinery that allows reactions to take place. Enzymes are large protein molecules that catalyze biochemical reactions by lowering activation energies. Enzymes accomplish catalysis by configuring chemical reactants in the best possible spatial arrangement for reactivity. This enzyme catalyzes the release of carbon dioxide when producing energy from carbohydrates. Combining with the coenzyme helps the inactive enzyme achieve the appropriate three-dimensional configuration. For this reaction, thiamine must be present as the coenzyme. The green coenzyme molecule above is thiamine pyrophosphate, the cofactor needed to permit the enzyme and the substrate, pyruvate, to combine in the correct orientation to catalyze the reaction. Now all three molecules are nested together, and the substrate, pyruvate, combines chemically with the coenzyme, thiamine. The biochemical reaction can take place and energy, as ATP, and CO2 are produced as products of the reaction. Once this happens, the remaining complex, containing the two-carbon fragment, thiamine, and the apoenzyme, rearranges and releases the two-carbon fragment, acetaldehyde, and the ATP. The departure of the acetaldehyde opened the thiamine pyrophosphate to combine with another molecule of pyruvate, and the cycle continues. Meanwhile, the energy is now in the molecule ATP and is available to do work or to be stored. Another way that a coenzyme can influence the reaction between substrate and enzyme is to cause a change in configuration of the enzyme without interacting with the substrate. Here you see the conformational change in the enzyme needed to configure the enzyme to accept the substrate in a lock and key complex. This is called an allosteric control. The cofactor approaches the inactive enzyme and bonds to it in an enzyme-coenzyme complex. The new configuration is a good fit for the substrate. Once this enzyme cofactor substrate complex is formed, energy can be released as ATP, and the other reaction products can be released from the complex. Now the correctly configured active site on the enzyme is free to engage another substrate. The amount of B vitamins that are needed to serve as coenzymes is quite small. There is enough thiamine activity in 1.2 milligrams to meet the average person's needs all day. Extra B vitamins will not produce more energy. Excess B vitamins in the body will pass out of the kidneys into the urine. This table shows some details about several of the roles played by B vitamins as coenzymes.
The optimum conditions that are needed for the enzymes. Okay, so all enzymes have an optimum temperature and pH at which they will work best. If they're not in these optimum conditions, what will happen is they degenerate or go through degeneration. This is the process modifying the molecular structure of a protein. It involves the breaking of many of the weak linkages or bonds, such as hydrogen bonds, and they change the shape of the active site, and the substrate will not be able to fit. Okay, the easy example of this is if you had a egg and you went to scramble it. Okay, in order to scramble the egg, okay, we have to add heat. Okay, after we add the heat, it's in a much different shape and cannot be changed back into the original form. That's what happens when an enzyme is denatured. also affected by the environment. Most enzymes function maximally in a narrow range of temperature and pH that we mentioned. With the pH, let's think of the stomach acid, okay, inside our gut. If it's too low, the enzymes can't work. And if it's too high, the enzymes cannot work as well. They change that shape much like the scrambled egg. To understand a normal enzyme, okay, we have that attachment at the active site. But with heat or a change in pH, it has now changed shape, and this is no longer able to fit into the enzyme.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Okay, a ribozyme is a ribonucleic acid RNA enzyme that catalyzes a chemical reaction. The ribozyme catalyzes specific reactions in a similar way to that of protein enzymes. Also called catalytic RNA, ribozymes are found in the ribosome where they join amino acids together to form protein chains. Ribozymes function as part of the large subunit ribosomal RNA to link amino acids during protein synthesis. Of metabolism, and will be okay. A metabolic pathway is a chemical reaction occurring in the metabolic pathway. Each step is coordinated by a specific enzyme. Okay, we have catabolic pathways, which break down cellular components, and these are exergonic reactions. And we have anabolic pathways, which synthesize cellular components, and these are endergonic reactions, which must be coupled to an exergonic reaction. Okay, so catabolic is breaking apart, anabolic is building. So think of anabolic steroids building muscle. That's an anabolic pathway. Reactions, breakdown of reactants. They are used for recycling building blocks. They are used for energy to drive endergonic reactions. So energy stored in intermediates, okay, such as ATP and NADH. An example is the breakdown of glucose into two molecules of pyruvate during glycolysis, and ATP is produced. Okay, now we haven't discussed glycolysis yet, but we have the breakdown of glucose into two molecules of pyruvate during glycolysis, which is basically breaking a 6-carbon sugar into two 3-carbon sugars. Okay, and this produces ATP, and we'll be talking about this in the next unit. Two ways to make ATP. Okay, we have a substrate level phosphorylation, which says enzymes directly transfer phosphate from one molecule to another molecule. And then we have chemosis, which is energy stored in an electrochemical gradient, which is used to make ATP from ADP and phosphate. Okay, I understand that this is a little confusing at the moment, but once again, we'll be going over this in next week's unit. reaction. Okay, so an electron removed from one molecule is added to another. We have what's oxidation, which is the removal of electrons, and reduction, which is the addition of electrons. And we have this formula, and it says A is oxidized and B is reduced. Now, this might be difficult to understand, but in any class and any chemistry or biology, the easy way to understand this is to remember the acronym LEO GER. Okay, so to easily understand it, LEO GER is a loss of electrons is oxidation, and a gain, sorry, GER, sorry, a gain of electrons is reduction. Okay, so as long as we remember Leo GER, that should be GER. Leo GER, we know a loss of electrons is oxidation and a gain of electrons is reduction. And as long as you remember that, you'll always know what oxidation and reduction means when talking in these processes. pH. Okay, electrons removed by oxidation of organic molecules are used to create energy intermediates like NADH. NAD plus, okay, what we have here is NADH useful in two ways. They releases a lot of energy when oxidized that can be used to make ATP, and they can donate electrons during synthesis reactions to energize them. Now, I understand that this sounds foreign. This is what we'll be discussing next week, but we have a slight introduction video so you can understand the processes coming.
Cells obtain energy during cellular respiration by oxidizing food molecules such as glucose. The energy derived from these oxidation reactions is used to form ATP. Oxidation can be defined as the removal of hydrogens from a molecule. Since a hydrogen consists of a proton and an electron, a proton and an electron are removed during oxidation. Whenever a molecule is oxidized, hydrogens removed, another molecule must be reduced, hydrogens added to it. During an oxidation-reduction reaction in the cell, an enzyme is involved in transferring the hydrogen, proton plus electron, to a coenzyme called NAD+. This enzyme has a binding site for both the substrate and NAD+. Once the substrate and NAD+ are bound with the enzyme, the hydrogen is transferred from the substrate to the NAD+. In other words, the substrate is oxidized, loses the hydrogen, and the NAD+ is reduced to NADH. As with all enzyme-mediated reactions, once the reaction is complete, the products separate from the enzyme and the enzyme can be used again. NADH, a high-energy electron carrier, diffuses away and is available to donate the hydrogen to other molecules.
Now, if you had difficulty understanding that video, you can go back and rewatch it and just think Leo GER the entire time and follow the electron, and you can understand the difference between oxidation and reduction reaction. Anabolic reaction. Okay, these are biosynthetic reactions. They make large macromolecules or smaller molecules not available from food. It requires energy inputs from those intermediates, such as NADH or ATP, to drive the reaction. Of those metabolic pathways, okay, we have gene regulation, which turn genes on and off. We have cellular regulation, which is cell signaling pathways like hormones. We have a biochemical regulation, which is feedback inhibition, okay, which is product of pathways inhibits early steps to prevent overaccumulation of a product. And we have regulation of the rate limiting or the slowest step. So if the rate limiting step is inhibited or enhanced, such changes will have the greatest influence on the pathway. Okay, so we have this diagram, which is, okay, so we have the initial substrate and intermediate one, intermediate step two, and then a final product. Okay, but to understand that easier, okay, I have two short videos for you as well.
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
Okay, so that was just to show you the example of the pathway.
Many of the enzyme-catalyzed reactions that occur in a cell, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, the product of one reaction becomes the substrate for the next reaction. If the end product of a pathway, such as an amino acid, becomes available in the environment, it is unnecessary and wasteful for the cells to continue to produce the product. Cells therefore have the ability to shut down a pathway when it is not needed. In feedback inhibition, the end product of the pathway reacts with the first enzyme that is unique to the pathway. The reaction occurs at a site on the enzyme that is different from the active site, called the allosteric site. When the product binds to the allosteric site, the enzyme undergoes a conformational change and can no longer react with its substrate. There is no substrate for subsequent steps in the pathway, and the final product is no longer synthesized.
And there you have your example of a regulation of the organic molecules. Okay, so most large molecules exist for a relatively short period of time. They have what's called a half-life, which it's the time it takes 50% of the molecules to be broken down and recycled. All living organisms must efficiently use and recycle these organic molecules. Expression of a genome allows cells to respond to changes in their environment. So RNA and proteins made when needed, okay, and they're broken down when they're not. What we call a proteasome. They are a large complex that break down proteins using protease enzymes. Okay, these protease enzymes cleave bonds between the amino acids. We have one called ubiquin, which tags target proteins to the proteasome to be broken down and recycled. Okay, and this ubiquin tagging allows the cell to degrade improperly folded proteins, rapidly degrade proteins to respond to changing cell conditions, and recycle amino acids for new proteins. And we'll be going over this enzyme when we go over electron transport chain. Here it shows it in a five-step process, which we have talked about already. Okay, lysosomes contain hydrolysases to break down proteins, carbohydrates, nucleic acids, and lipids. So we know lyse is to break, so lysosomes are to break these different molecules apart. They digest substances taken up by endocytosis. Okay, and we have autophagy, which is recycling worn-out organelles using an autophagosome. Okay, and this is an example is the immune the immune cell going around and engulfing a bacteria or a virus to eat it and destroy it. In this example, a couple questions. First is which of the following is probably the enzyme for the substrate amylose? And that would be amylase. So what's to understand this question is the first prefix part of the word normally matches, okay, the enzyme to the substrate. That's the optimum range for a protease used in digestion. So what do you think the pH would be? Okay, if we're talking digestion, the pH would be most optimally 1.5 because we're talking about very strong acid inside the stomach. What would we think the temperature would be? Okay, the body functions at a degree of 98.6 at all times for its best functioning. Okay, the reason we get a fever is because our body is trying to kill bacteria and viruses by denaturing them. Now, this is the problem. If the fever goes too long, it can also start denaturing our own systems. Okay, but this is why we optimally want to maintain a 98.6 degrees. The person stops making stomach acid and the pH of the stomach becomes closer to a pH of 4. Okay, the enzymes will not work as well. It's that simple because they've denatured. How to spot an enzyme? Okay, it usually ends in the suffix ase or ace. And examples of that is catalase, amylase, lactase, or protease. It's probably the enzyme that breaks down protein. Good, if you went with that prefix, you chose protease. Uh, Introduce a little bit of homeostasis. What homeostasis is the ability of the cell to maintain stable internal conditions by adjusting to changes inside and outside the cell. And we have examples of sweating, shivering, urinating, breathing. Okay, but to understand this a little bit better, okay, want you to think of, say it's your birthday, okay, and someone gives you a whole cake. Okay, these are, a cake with candles on it. Okay, and so you decide, instead of just having one piece of cake, you eat that entire cake. Okay, what happens in your body is your body increases in sugar. Okay, as a response to that sugar, your liver, okay, and pancreas, sorry, your pancreas will produce insulin to bring the sugar back down to normal. Okay, but then you got really, really depressed for eating this cake. So you decide not to eat for an entire 2 weeks. Okay, because you didn't eat for that 2 weeks, your sugar has gone low. Okay, and your body produces, the liver produces a chemical called glucagon to bring the sugar back up to this normal range. So you body wants to remain at this level perfect condition at all times, but it never really does for what we bring into the environment. Okay, but this happens with our body temperature, with digestion. Every process follows this homeostasis rule. Enzymes that contribute to homeostasis, they allow the body to perform reactions that would not normally happen on their own. Okay, they allow the body to perform reactions that make the process happen quicker than it would on its own. Die. Okay, digestion relies on enzymes so we can use and absorb nutrients from our food. So if we did not have digestive enzymes, the food that we ate would rot inside our bodies and we would starve long before the nutrients naturally broke down for use in our cells. For example, we have lactose sugar is found in milk. What would be a good explanation for why a person would be lactose intolerant? Okay, very simply, they are unable to make the lactase enzyme. The buildup of undigested sugar causes them to become bloated. So that sugar is actually in a person's gut rotting. Okay, that's lactose intolerant. Two solutions that we could have for this problem. Okay, we have take the lactose sugar out of the milk or simply don't eat dairy, or give the person a lactase enzyme to help them digest it. And this is where the products such as lactate come into play. Okay, here we have milk that has the lactose sugar taken out, as well as the enzyme that can be taken in pill form before you eat dairy. Increasing the amount of enzyme speed up a reaction. The answer to this is yes. Okay, increasing the amount of enzyme increases the number of active sites. The more active sites we have, the quicker the reaction. The amount of substrate speed up a reaction. And the answer to this is no. Okay, if the active sites of the enzyme are all full, there is nowhere else for the substrate to bind to the enzyme. The substrate will wait in solution until more active sites are free to bond. This is what happens with the person that's lactose intolerant. They have very few enzymes to digest that sugar, and the rest of the sugars just sit there and wait until the enzymes are recycled and can be used up again. Once again, don't forget that if the videos were too fast for you, you can go back and find them in the animation sections on Canvas. And good luck on the quiz.